At 200 meters below the surface of the ocean, something happens that most people have never thought about, and that changes everything. The light disappears. Not gradually, not in a slow fade that gives you time to adjust, but in stages so precise that scientists have mapped them, named them, and used them to divide the ocean into zones as distinct from each other as different planets. The red wavelengths go first, absorbed within the first few meters of seawater, then orange, then yellow. By 100 meters, the world is blue and dim. By 200 meters, the blue is gone too. What remains below that boundary is not simply darkness. It is the most extensive dark environment on Earth, a volume of cold water so vast that it dwarfs every other habitat on the planet combined, home to creatures so strange and so perfectly adapted to conditions that would kill almost anything else that we have been studying them for over a century and still do not fully understand what we are looking at. This is the midnight zone, and most of it has never been seen by human eyes. If you could dive into the midnight zone tonight and keep going, how far do you think you would travel before hitting bottom? Tell us in the comments. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. Clear in the past few decades, as technology has finally allowed us to visit the deeper layers directly rather than infer their existence from instruments lowered on cables. The topmost layer, the sunlit zone or epipelagic zone, extends from the surface to approximately 200 meters. This is the layer that most people mean when they think of the ocean. The warm, bright, productive zone where phytoplankton photosynthesize, where fish school in the millions, where the food webs that ultimately sustain almost all ocean life begin. It represents roughly 2% of the total volume of the ocean. Everything below it is something else entirely. Below the sunlit zone, from 200 to approximately 1,000 meters, lies the mesopelagic zone, the twilight zone. This is the layer where what little light remains from above filters down as a dim blue glow, too faint for photosynthesis, but sufficient for vision, sufficient to create a world where seeing and being seen still matters. The organisms of the twilight zone have eyes calibrated to detect the last traces of downwelling light, eyes that can distinguish the silhouette of a predator above against the faint blue background, eyes that have been refined by millions of years of evolution in exactly this quality of light. The twilight zone is also the zone of the great daily vertical migration, one of the largest movements of animal biomass on Earth, in which billions of individual organisms ascend toward the surface each evening to feed in the relatively food-rich shallows and descend again before dawn to the safety of the dark. The twilight zone is not a transition. It is a world. Below 1,000 meters, the last traces of sunlight are gone, permanently and absolutely. At 1,000 meters and below, no photon from the sun has ever arrived. The darkness is not the darkness of a room with the lights out, where ambient light creeps under doors and around curtains. It is not the darkness of a moonless night, where clouds glow faintly with reflected light from distant cities. It is something that has no analog in the surface experience of a human being, the complete and permanent absence of light in any form except what living organisms produce themselves. This is the bathypelagic zone, beginning at 1,000 meters and extending to approximately 4,000 meters. It is called the midnight zone, and it contains, by volume, more habitable space than all the sunlit and twilight zones combined, more habitable space than all terrestrial environments on Earth put together, more habitable space than anything else on this planet. It is the largest living space on Earth, and we know almost nothing about it. The physical conditions of the midnight zone are extreme by any surface world standard, though the organisms that have evolved to live there experience them as simply normal. The temperature in the bathypelagic zone ranges from approximately 4 degrees Celsius near the top to near freezing at the bottom, depending on location. This cold is not the cold of a winter day which a human body can adapt to with appropriate clothing and shelter. It is the cold of water that has been circulating in the deep ocean for decades or centuries without returning to the surface. Water that carries the thermal memory of its origin in the cold polar regions where it sank and began its long journey along the ocean floor. The pressure in the midnight zone ranges from approximately 100 atmospheres at 1,000 meters to roughly 400 atmospheres at 4,000 meters. 1 atmosphere is the pressure you are experiencing right now, the weight of the entire column of air above you pressing down on your body. 400 atmospheres is 400 times that. It is a pressure that would instantly crush an unprotected human body, that would collapse the gas-filled spaces in the lungs and sinuses and middle ear, that would force nitrogen and other gases into the bloodstream in concentrations that would be immediately fatal. For the organisms that live in the midnight zone, this pressure is the medium in which they exist. They have been shaped by it so completely that many of them would be unable to survive at the surface, where the pressure would be catastrophically low by their standards. The history of human attempts to understand the midnight zone is a history of technological limitation gradually overcome. For most of human history, the deep ocean was simply unknown. The surface was navigable. The shallow coastal waters were fishable, but what lay below was entirely beyond reach, beyond knowledge, beyond imagination, except in the form of myth. The monsters that populated the edges of medieval maps, the sea serpents and leviathans of maritime legend, were not mere fantasy. They were the visual language of genuine ignorance. Nobody knew what was down there. The mythology filled the gap. The first systematic attempt to probe the deep ocean came with the HMS Challenger expedition of 1872 to 1876, the voyage that effectively founded the science of oceanography. The Challenger's crew lowered weighted lines to the seafloor and brought up samples of sediment and organisms from depths that no one had believed were inhabited. They found life at 4,000 and 5,000 meters. Life in the darkness, in the cold, under pressures that their instruments barely registered, but that would have been lethal to any of them. The discovery that the deep ocean was not a lifeless void was the most significant biological finding of the 19th century, and it has been generating surprises ever since. The instruments that followed the Challenger's weighted lines were progressively more sophisticated, Echo sounders in the early 20th century allowed ships to measure depth by timing the return of sound pulses from the seafloor, replacing the slow and inexact process of lowering ropes with a technology that could map the topography of the deep ocean from the surface. Trawl nets were designed to fish at depth, bringing up specimens of midnight zone organisms that had never been seen before. Though the process of hauling them to the surface killed most of them and damaged many beyond recognition, the pressure change was simply too great. An organism adapted to 200 atmospheres, brought rapidly to the surface, experiences something analogous to explosive decompression in reverse. The gases dissolved in its tissues under pressure come out of solution too fast. The structures that maintain their shape under extreme compression fail under the relatively low pressure of the surface. Many of the first specimens collected from the midnight zone arrived in a condition that made them difficult to identify, let alone understand. The development of deep-sea submersibles in the mid-20th century changed the terms of midnight zone research fundamentally. For the first time, human beings could descend into the dark and observe its inhabitants in their actual environment, alive, behaving normally, unaware that they were being watched. The first deep dives revealed immediately that the midnight zone was nothing like what surface observations had suggested. The organisms collected in trawl nets had given a misleading picture, sampling only those species slow enough or unfortunate enough to be caught, missing the faster swimmers entirely, and presenting everything in a condition of damage and death that made behavioral observation impossible. The submersible view was different. The midnight zone, seen from inside a pressurized sphere descending through it, was dark in every direction except for the cone of light produced by the submersible's own powerful lamps. And in that cone of light, the organisms appeared, strange, improbable, moving with a slowness and deliberateness that reflected the low metabolic rates of cold water animals and the scarcity of food that made every movement an energy budget calculation. They were not monsters. They were not the terrors of the deep that mythology had promised. They were something stranger than monsters. Organisms so exquisitely adapted to their specific environment that they seemed to belong to a different biological reality than anything at the surface. The midnight zone is divided informally by oceanographers into subzones based on depth and temperature, though the boundaries are not sharp and vary by location. The upper midnight zone, roughly 1,000 to 2,000 meters, is where the influence of the surface world is still weakly felt. The chemistry of the water at this depth retains some connection to the surface through the slow circulation of deep water masses. The food supply, while limited, is somewhat better than in the deeper parts of the zone because marine snow, the constant gentle fall of organic particles from the layers above, has not yet been fully consumed by the organisms of the mesopelagic zone. The lower midnight zone, from 2,000 meters to the transition into the abyssal zone at approximately 4,000 meters, is more isolated. The food supply is sparser. The temperature is lower. The pressure is higher. The organisms that live here have had to develop more extreme adaptations to the same fundamental challenges. How to find food in the dark, how to find mates in the dark, how to avoid predators in the dark, and how to maintain the biochemical machinery of life at temperatures and pressures that would shut down most biological processes. What the midnight zone lacks in light, it compensates for, in part, with chemistry. The water at these depths is rich in dissolved minerals and gases that have accumulated over the centuries during which it has been out of contact with the surface and the atmosphere. Deep water is generally oxygen depleted compared to surface water because the photosynthesis that produces oxygen occurs only in the sunlit zone, and the deep water's oxygen was consumed long ago by the respiration of organisms and the oxidation of organic matter. In some regions, at intermediate depths, there are pronounced oxygen minimum zones where the oxygen content drops to levels that would asphyxiate most surface organisms. The creatures that can tolerate these oxygen minimum zones, or even exploit them as refuges from predators that cannot, represent some of the most physiologically unusual organisms in the ocean. Below the oxygen minimum zones, at greater depths, oxygen levels partially recover as the cold, dense water that has circulated from the polar regions brings dissolved oxygen from the surface into the deep. The distribution of oxygen in the midnight zone is not uniform. It is a complex, three-dimensional landscape, varying by depth, by latitude, by proximity to ocean ridges and seamounts, and by the history of the water mass at any given location. The sound environment of the midnight zone is one of its most striking and least appreciated features. Sound travels faster in water than in air, approximately 1,500 meters per second compared to 340 meters per second in air, and it travels much farther. The deep sound channel, also known as the SOFAR channel, it is a layer of water at approximately 600 to 1,000 meters depth, depending on location where the speed of sound reaches a minimum, and sound waves are focused and trapped, allowing them to propagate for thousands of kilometers with minimal loss of energy. Sounds produced in or near the SOFAR channel, including the low-frequency calls of baleen whales, the rumble of distant earthquakes, and the acoustic signatures of underwater explosions, can be detected by sensitive hydrophones on the other side of the ocean. The ocean is not silent. The midnight zone is filled with sound. The clicks of sperm whales hunting at depth, the groans and creaks of the seafloor under tectonic stress, the biological crackling of shrimp colonies on seamounts, the low-frequency moans of fin whales communicating across ocean basins. For the organisms that live in the midnight zone, sound is often more important than vision for navigation, communication, and prey detection. And the ocean, at midnight zone depths, is acoustically one of the richest environments on Earth, even as it is optically one of the darkest. The transition from the midnight zone to the abyssal zone below it, at approximately 4,000 meters, is marked by a gradual change in the character of the environment rather than a sharp boundary. The abyssal zone is colder, darker, and more uniformly hostile than the midnight zone, if such a comparison is meaningful among environments where all of them would instantly kill a surface organism. The sediment-covered abyssal plain that underlies most of the midnight zone's water column has its own biology, its own chemistry, its own geological history, a system that extends the midnight zone's darkness and cold and pressure to include the seafloor itself. What lives on and in that sediment, and what the relationship is between the water column of the midnight zone and the seafloor below it, is a question that connects the biology of the dark water to the geology of the ocean basin, and that has implications for understanding the ocean as a system rather than as a collection of separate environments. The midnight zone is not merely a layer of dark water. It is the largest connected habitat on Earth, linking the productive surface to the ancient sediment below through a column of cold, dark, pressurized water that has been sustaining life for as long as the ocean has existed. And what lives in that column and how it lives is where the story of the midnight zone really begins. The scale of the midnight zone is worth dwelling on before we descend further into its specific biology and physics. The ocean covers approximately 71% of the Earth's surface. Of that ocean, roughly 75% is deeper than 1,000 meters, which is to say, within or below the midnight zone. The total volume of water in the midnight zone and the abyssal zone combined is approximately 1.335 billion cubic kilometers. The total volume of all terrestrial environments on Earth, every forest and grassland and desert and mountain range and river and lake, is a small fraction of that number. When we say that the midnight zone is the largest living space on Earth, this is not a rhetorical flourish. It is a straightforward mathematical fact. The Earth is, in terms of habitable volume, primarily an ocean planet, and the ocean is primarily a dark, cold, pressurized environment that most people have never visited or meaningfully thought about. The midnight zone is not the exotic edge of the biosphere. It is the biosphere's dominant component. Everything else, every forest and prairie and coastal reef and tidal pool, is the margin. There's one more physical property of the midnight zone that belongs in this opening account, one that is less dramatic than the pressure or the cold, but that shapes everything about how life operates at these depths. Time. The midnight zone operates on timescales that have no equivalent at the surface. The water circulating through the midnight zone has been out of contact with the atmosphere for decades to centuries. The organisms that live there grow slowly, reproduce rarely, and live long. The processes that cycle nutrients through the system operate over years and decades, rather than days and weeks. Events at the surface, a bloom of phytoplankton, the death of a large whale, a change in surface temperature, take months or years to propagate their effects to midnight zone depths. The midnight zone is insulated from the surface world not just by kilometers of water, but by the time it takes for any signal from above to arrive. This insulation is not complete. The vertical migration of mesopelagic organisms connects the zones every night. The marine snow falls continuously. The great circulation of the deep ocean connects the midnight zone to the surface on scales of centuries. But the dominant experience of the midnight zone for the organisms that live there and for the physical and chemical systems that sustain them is one of slowness, of patience, of processes unfolding on time scales that make a human lifetime seem brief. The midnight zone is not waiting for something to happen. It is already happening, everywhere, all the time, in the dark, at a pace that the surface world does not recognize as urgency, but that the midnight zone has been perfecting for millions of years. In the midnight zone, light is not absent. It is produced. This distinction matters more than it might initially seem. The absolute darkness of 1,000 meters and below is not a void of all electromagnetic radiation. It is a void of solar radiation, of the light that originates 150 million kilometers away and drives the biology of the surface world. But within that darkness, living organisms produce their own light through a chemical process called bioluminescence, and they produce it in quantities and varieties that make the midnight zone paradoxically one of the most visually complex environments on Earth. Estimates vary by method and by ocean region, but the most careful surveys suggest that somewhere between 70 and 90% of all organisms in the midnight zone are capable of producing light. Not incidentally, not as a side effect of other chemistry, as a primary biological capability, as essential to their survival as their ability to find food or avoid predators or reproduce. Bioluminescence in the midnight zone is not a curiosity. It is the medium of communication, predation, defense, and reproduction in a world where sunlight has never existed. The chemistry of bioluminescence is, at its core, simple. A molecule called luciferin reacts with oxygen in the presence of an enzyme called luciferase to produce light. The reaction is exothermic, but produces almost no heat, which is why bioluminescence is often called cold light. Nearly all of the chemical energy is converted directly into photons, with minimal energy lost as heat. This efficiency vastly exceeds anything human engineers have achieved with electric lighting. The LED, which is the most energy-efficient artificial light source currently in widespread use, converts roughly 50 to 60% of its electrical energy into visible light. The bioluminescent reaction converts chemical energy into light with an efficiency that significantly exceeds anything human engineering has matched in a comparable package. Evolution, working on this problem for hundreds of millions of years, arrived at a solution that human technology is still attempting to match. What makes bioluminescence in the midnight zone so biologically extraordinary is not just the chemistry, but the variety of its applications. Different species have evolved to use bioluminescence for fundamentally different purposes, and the diversity of those purposes mirrors the diversity of the ecological challenges that organisms face in the dark. The anglerfish uses bioluminescence as a lure. The female of most anglerfish species carries a modified dorsal spine on her head, a structure called an esca that contains bioluminescent bacteria living in a symbiotic relationship with the fish. The bacteria produce light continuously, and the fish uses this light to attract prey in the darkness. The light is not produced by the fish itself. It is produced by microorganisms that live inside the lure, receiving nutrients from the fish's blood supply in exchange for the light that the fish cannot produce on its own. This is not a simple relationship. It is a co-evolutionary partnership between two organisms that have become so dependent on each other that neither can function without the other. The bioluminescent bacteria in the anglerfish's lure have, in many species, lost the ability to produce light independently when removed from the lure environment. The lure itself degenerates without the bacteria. The partnership is obligate and mutual, two organisms functioning as one because neither can survive alone. The dragonfish in contrast, uses bioluminescence as a weapon rather than a lure, and in a way that is more sophisticated than almost anything else in the Midnight Zone's biological repertoire. Most bioluminescent organisms in the deep ocean produce blue or blue-green light for the straightforward reason that blue light penetrates seawater more efficiently than any other wavelength and is therefore the color most easily detected by eyes adapted to the deep ocean. Nearly every deep-sea animal's visual system is tuned to blue-green wavelengths. This is the color of bioluminescence that most deep-sea eyes can see. The dragonfish, specifically several species of the genus Malacosteus and related genera, produces light in the far-red wavelength at approximately 700 nanometers. Far-red light is essentially invisible to most deep-sea organisms whose photoreceptors are tuned to blue-green. The dragonfish is illuminating its environment with a searchlight that its prey cannot see. It can see them because it has evolved photoreceptors sensitive to its own far-red wavelength. They cannot see it because their visual systems have been shaped by an environment in which far-red light has never, until the dragonfish evolved to produce it, existed. The dragonfish's bioluminescence is a private frequency, a communication channel to which only the dragonfish has access. It is one of the most elegant examples of evolutionary innovation in the deep ocean. Counter-illumination is another use of bioluminescence that is widespread in the Midnight Zone, and that illustrates the sophistication of the evolutionary solutions that darkness and predation pressure have produced. Many mesopelagic and bathypelagic fish and squid carry rows of small light-producing organs called photophores along their bellies. These photophores produce a faint downwelling light that matches the dim bioluminescent glow filtering from above, effectively canceling the fish's silhouette as seen from below. A predator, looking upward from deeper water, sees other animals as dark shapes against a faintly lighter background. An animal with counter-illumination photophores eliminates this silhouette, making itself invisible to predators approaching from below. The light produced by the photophores is not random. It is calibrated to match the intensity and color of the downwelling light at any given depth, and some species have photoreceptors that measure the ambient light above them and adjust their photophore output accordingly in real time. They are, in effect, carrying a continuously self-adjusting camouflage system tuned to the specific lighting conditions of their current depth. The sophistication of this system exceeds anything human camouflage technology has achieved for underwater applications. The hatchetfish, family Sternoptechidae, is perhaps the best-known example of a counter-illumination specialist. These small, deep-bodied fish, typically 2 to 6 centimeters long, have extremely compressed, laterally flattened bodies with reflective silver scales that reduce their side profile to near invisibility from front and rear angles. Their large, upward-directed eyes are specialized for detecting prey and predators against the dim, downwelling light above. And along their entire ventral surface, they carry rows of photophores, producing the precisely calibrated, downwelling light that makes them invisible from below. The hatchetfish is not a particularly dramatic organism by the standards of the Midnight Zone. It is small, and it eats zooplankton, But the engineering embedded in its body plan, the combination of lateral compression, reflective scaling, upward vision, and counter-illumination bioluminescence, is the product of an evolutionary process that has been solving the problem of surviving in dim, predator-rich water for tens of millions of years. The solution is more sophisticated than anything we have designed for comparable purposes. The siphonophore represents bioluminescence at a scale and in a form that has no equivalent in any other group of organisms. Siphonophores are colonial animals composed of individual units called zooids that are genetically identical but specialized for different functions within the colony. Some zooids handle propulsion, others handle feeding, others handle reproduction, others provide structural support. The colony as a whole behaves as a single organism, but it is in fact an assemblage of individuals so deeply integrated that the distinction between individual and colony becomes difficult to maintain. The largest siphonophores, including Praia dubia, can reach lengths of 40 to 50 meters, making them among the longest animals ever recorded on Earth. And many siphonophore species are bioluminescent, using light for purposes that range from communication between zooids within the colony to the attraction of prey. Some siphonophores produce bioluminescent flashes so bright that they can be seen by the naked eye from a distance of several meters in the dark water of the midnight zone, a scale of bioluminescence that would be startling even if you were expecting it. The siphonophore is not a small curiosity of the deep. It is one of the largest and most architecturally complex organisms on Earth, and it glows. The use of bioluminescence for communication and mate finding in the midnight zone is less well understood than its use for predation and defense, partly because observing social behavior in deep-sea organisms requires sustained observation under conditions that are technically challenging, and partly because many of the relevant behaviors occur in the open water column at depths and distances that make observation difficult even with the best current submersible technology. What is known is that many bioluminescent species produce light in patterns that appear to be species-specific and sex-specific, suggesting that the light serves a role in species recognition and mate selection, analogous to the role of visual signals in surface animals. Some squid species flash complex bioluminescent patterns across their skin using chromatophores and photophores in combination, patterns that vary with behavioral context in ways that researchers are still attempting to decode. The communication happening in the midnight zone, in light that human eyes would need instruments to detect, and that neither party to the communication intends for outside observation, is a language we're only beginning to learn the existence of. The bacterial component of bioluminescence in the midnight zone deserves its own attention because it connects the individual light production of organisms like the anglerfish to a broader ecological and evolutionary story. Bioluminescent bacteria are ubiquitous in the ocean, present in the water column, in the guts of fish, in the tissues of specialized light organs, and on the surfaces of decaying organic matter. They are among the most ancient producers of bioluminescence on Earth, with the luciferin luciferase chemistry appearing in bacterial lineages that diverged from other bacteria billions of years ago, before the Cambrian explosion, and before the evolution of animals with eyes to see the light they produce. The bioluminescence of complex organisms like fish and squid is, in many cases, ultimately derived from bacterial chemistry, either through direct symbiosis, as in the anglerfish, or through the evolution of similar chemistry in animal cells, through a process that biologists are still working to trace. The light of the midnight zone, in this sense, is ancient light, chemistry that predates the eyes that evolved to detect it by hundreds of millions of years. The darkness that the light illuminates is younger than the light itself. The barrel eye fish, Macropina microstoma, represents a different approach to vision in the midnight zone, one that does not rely on bioluminescence at all, but instead pushes the limits of what a vertebrate eye can do with vanishingly small quantities of ambient light. The barrel eye is a small fish, typically 15 centimeters long, that lives in the mesopelagic and upper bathypelagic zones of the Pacific Ocean. Its defining feature is a pair of eyes that point directly upward within a transparent, fluid-filled dome that covers the top of its head. These eyes are not fixed in place. They can rotate within the dome, moving from an upward orientation to a forward orientation when the fish detects prey and pivots to capture it. The barrel-shaped eyes, from which the fish takes its name, contain extremely large lenses and very dense concentrations of rod photoreceptors, giving the barrel eye exceptional sensitivity to dim downwelling light. The fish spends most of its time hovering almost motionless in the water column, its upward-pointing eyes scanning the faint illumination above for the silhouettes of small crustaceans and other prey. The transparent dome covering the barrel eye's eyes was unknown to science until relatively recently. Earlier specimens, collected in trawl nets and examined at the surface, always arrived with the dome collapsed or missing, destroyed by the pressure change during ascent. Scientists examining these specimens assumed from the structure of the skull that the eyes pointed forward, as in most fish. It was only when ROV footage captured living barrel eyes in their actual environment that the transparent dome became apparent, revealing a body plan that had been completely misunderstood for decades. The fish that scientists thought they knew turned out to be a fish they had never actually seen correctly. The midnight zone has a long history of revealing that our assumptions about what lives there, based on damaged specimens hauled to the surface, are wrong in fundamental ways. Every time we have been able to observe these organisms alive, in place, we have found something different from what the trawl net samples suggested. The vampire squid, Vampiroteuthis infernalis, occupies a taxonomic position as unusual as its appearance. It is not a true squid and not a true octopus. It belongs to its own order, Vampiromorphidae, a group with no other living members, an evolutionary survivor from a lineage that diverged from both squid and octopus hundreds of millions of years ago. It lives in the oxygen minimum zones of the world's oceans, at depths of 600 to 900 meters, in water so depleted of oxygen that most predators cannot function there. This oxygen refuge is its primary defense. The vampire squid can tolerate oxygen concentrations of approximately 3% of saturation, a level at which most marine animals would suffocate within minutes. In this nearly anoxic environment, it drifts slowly, trailing its web-like arms, collecting the marine snow and organic particles that drift through its zone. It does not hunt. it gathers. And it is bioluminescent throughout its body, capable of producing light from photophores distributed across its entire surface, from the tips of its arms to the underside of its webbing, in patterns that researchers believe are used for communication and possibly for disorienting predators that do manage to follow it into the oxygen minimum zone. The vampire squid is not merely a deep-sea animal. It is a living fossil, a representative of a biological lineage that has been hiding in the oxygen minimum zones of the ocean while the rest of the cephalopod world evolved around it, surviving by occupying an environment too hostile for most of its competitors. The anglerfish deserves more than a mention of its lure. There are more than 200 described species of anglerfish in the deep ocean, and their biology is among the most extreme in the midnight zone. The female of most deep-sea anglerfish species is the predator, the large animal with the bioluminescent lure, the massive mouth, the teeth that curve inward like fish hooks, the expandable stomach that can accommodate prey larger than the anglerfish itself. The male is something different. In most deep-sea anglerfish species, the male is dramatically smaller than the female, sometimes 100-fold or more. The male has no functional digestive system. What he does have are highly developed nostrils and eyes, designed for a single purpose: finding a female in the darkness. When a male locates a female, attracted by the chemical pheromones she releases and guided by the light of her lure, he bites into her skin. Over time, their circulatory systems fuse. The male's body degenerates, losing everything except the reproductive organs that the female needs access to. He becomes, functionally, a parasitic appendage, receiving nutrients from the female's bloodstream while providing sperm on demand. In some species, females have been found carrying 6 or more attached males simultaneously. The male anglerfish has solved the problem of finding a mate in the vast, dark, food-scarce midnight zone by ensuring that, once found, the connection is permanent. It is not a strategy that appeals to any human sense of individual dignity. It is a strategy that works perfectly and completely in the environment that produced it. The midnight zone does not produce organisms that are merely strange. It produces organisms that are strange in ways that are precisely, elegantly calibrated to the exact conditions of their environment. The strangeness is not incidental. It is the point. What bioluminescence ultimately reveals about the midnight zone is something that goes beyond the biology of individual species. It reveals that the darkness of the midnight zone is not an absence. It is a presence, a medium, a physical property of the environment that has been as formative as temperature or pressure in shaping every organism that lives there. Surface organisms evolve in a world where the primary sensory medium is sunlight. The organisms of the midnight zone evolved in a world where the primary visual medium is bioluminescence, where the patterns of cold blue-green light carry meaning, encoded by millions of years of coevolution between the producers and the detectors of that light. The visual ecology of the midnight zone is not a diminished version of surface visual ecology. It is a different visual ecology, built on different physics, shaped by different selection pressures, producing solutions that are in many respects more sophisticated than anything the sunlit world has generated. The darkness was a more demanding teacher than the light, and the midnight zone's organisms did not merely survive it, they turned it into a language. What that language is actually saying to the other organisms listening in the dark is a question that the next descent begins to answer. The midnight zone is not a passive environment. It is not a dark, cold tank of water in which organisms simply exist, waiting for food to arrive. It is an active ecological arena in which predators and prey are locked in an ongoing evolutionary arms race, in which the strategies for finding and killing are matched against the strategies for hiding and escaping, in which the physics of the environment, the darkness, the pressure, the cold, the scarcity of food, shape every weapon and every defense that evolution has produced. The hunters of the midnight zone have solved the problem of predation in conditions that should make predation nearly impossible in a three-dimensional environment with no hiding places except darkness itself, with prey that may be dispersed over cubic kilometers of open water, in a world where every expenditure of energy must be justified by a return. The solutions they have found are, without exception, extraordinary. The sperm whale is the largest predator that regularly descends into the midnight zone, and its hunting strategy represents one of the most impressive feats of biological engineering in the ocean. Sperm whales can reach lengths of 18 meters and weights of 57 tons. They breathe air, which means that every dive into the midnight zone is a breath hold dive, a plunge into cold and pressure on a single lungful of oxygen that must sustain the animal for the duration of the hunt and the return to the surface. Sperm whales have been recorded diving to depths of at least 2,200 meters, possibly deeper. They remain submerged for up to 90 minutes on a single breath. The physiology that makes this possible is a masterpiece of adaptation. The sperm whale's blood can carry oxygen at concentrations that would be toxic to a human. Its muscles are so saturated with myoglobin, the protein that stores oxygen in muscle tissue, that they are nearly black. Its rib cage can collapse completely under the pressure of depth without injury, allowing the lungs to compress without trapping air that would cause decompression sickness on ascent. Its heart rate slows dramatically during a dive, reducing oxygen consumption and allowing the stored oxygen to last longer. And its spermaceti organ, the massive waxy structure that fills much of its enormous head, may function as a buoyancy control device, changing density by regulating temperature through blood flow to help the whale sink or rise with minimal muscular effort. What the sperm whale hunts in the midnight zone is primarily the giant squid and the colossal squid, the largest invertebrates on Earth. This hunt between the largest toothed predator and the largest invertebrate has never been directly observed by human beings. It is one of the most consequential ecological interactions in the ocean, repeated countless times every day in the darkness of the midnight zone, and we have never once seen it happen. What we know of it comes from evidence. The stomach contents of sperm whales, which regularly contain squid beaks, sometimes in quantities of thousands. The scars on the skin of sperm whales, circular marks left by the suckers of giant and colossal squid, suggesting that the prey does not submit without resistance, and acoustic recordings of sperm whale click patterns at depth, which change in ways consistent with active echolocation-based hunting. The picture that emerges is of an encounter between two extraordinary animals, each the product of millions of years of evolutionary pressure in the midnight zone, each equipped with capabilities that have no equivalent at the surface. The outcome of any individual encounter is unknown. The aggregate outcome across the millions of dives that sperm whales make every year is that the sperm whale obtains enough food to sustain a body of 57 tons. The squid, collectively, obtain enough escapes to maintain their populations. The balance has been holding for millions of years. The giant squid, Architeuthis dux, is the prey in this equation, but it is not a passive prey. It is an active predator in its own right, hunting fish and smaller squid in the midnight zone with eyes that are the largest of any animal on Earth, reaching 30 centimeters in diameter in the largest specimens. The size of those eyes is the product of the same evolutionary pressure that produced the sperm whale's echolocation, the need to detect a predator in the dark before the predator detects you. A larger eye collects more photons, allowing it to resolve images at lower light levels. The giant squid's eye is at 30 centimeters, probably at the physical limit of what is useful for detecting bioluminescent light in the midnight zone. Larger would provide no additional benefit. The squid is using every photon available to it. And what it is looking for, specifically, is the bioluminescent glow that a sperm whale's passage through the bioluminescent organisms of the midnight zone produces, a bow wave of disturbed light that precedes the whale by potentially hundreds of meters, giving the squid time to attempt an escape. The giant squid's enormous eyes are, in this interpretation, not primarily for hunting, but for surviving being hunted. They are an anti-sperm whale adaptation, a sensor tuned to detect the specific signature that the largest predator in the midnight zone produces as it moves through the dark water. The viperfish, Chaliotis sloani, is a predatory fish of the mesopelagic and upper bathypelagic zones that represents the midnight zone's approach to teeth at its most extreme. The viperfish is typically 25 to 35 centimeters long, a modest size by any standard, but its fangs are so disproportionately long, so dramatically out of scale with the rest of its body, that it cannot close its mouth when they are in their normal position. The lower jaw teeth curve upward past the eyes when the mouth is closed. To compensate, the skull is hinged to rotate backward by nearly 90 degrees, allowing the mouth to open wide enough to swallow prey that approaches the viperfish's own body size. Along its belly, the viperfish carries a row of photophores that produce a downwelling light matching the ambient illumination above, providing the same counter-illumination camouflage used by the hatchetfish. On its dorsal fin, a modified spine carries a small bioluminescent lure that attracts prey to within striking distance. The viperfish is therefore both a counter-illuminated prey and a bioluminescent predator simultaneously, a dual function that reflects the dual reality of every organism in the midnight zone. Everything is both predator and prey, and the same adaptations that help it avoid being eaten help it eat. The stoplight loosejaw, Malacosteus niger, is perhaps the most conceptually strange predator in the midnight zone, and the most instructive about how the arms race between predator and prey in the dark operates. Like the dragonfish, the stoplight loosejaw produces far-red bioluminescence that is invisible to most deep-sea organisms. Unlike the dragonfish, the stoplight loosejaw has also lost the floor of its mouth and the membranes connecting its lower jaw to the rest of its head. Its lower jaw is essentially free-hanging, a bare skeletal structure with teeth and nothing else, hinged at the back and otherwise unconnected to the body. This means that the fish can open its mouth to an extraordinary angle, essentially unhinging its jaw to engulf prey. But why the loss of the membrane? The answer appears to be hydrodynamics. A fish with a membrane connecting its lower jaw to its body creates a bow wave when it lunges, a pressure wave that prey can detect through their lateral line, a sensory system that detects changes in water pressure and allows fish and other aquatic organisms to sense nearby movement. A fish with no membrane, lunging through the water with an open skeletal lower jaw, creates a minimal bow wave. The prey cannot detect it coming. The stoplight loosejaw is not only using an invisible searchlight, it is also invisible to the pressure-sensing system of its prey. It has eliminated 2 of the 3 ways its prey might detect its approach, leaving only the visual detection of its body, which is dark, in water that is dark. The black swallower, Chasmodon niger, has taken the midnight zone's food scarcity problem and addressed it through a solution so extreme that it occasionally kills the fish that employs it. The black swallower is a small fish, typically 25 centimeters long. It has a stomach that can expand to hold prey more than twice its own length and 10 times its own mass. The stomach is not merely large, it is extensible, capable of expanding to accommodate prey that is physically larger than the swallower's entire body. This is accomplished through a stomach wall made of highly elastic tissue and a skin that can stretch to allow the abdomen to bulge enormously when the stomach is full. The black swallower does not hunt for small prey items that fit conveniently within its normal body dimensions. It hunts for the largest prey item it can overpower, because in the food-scarce midnight zone, a single large meal may represent enough calories to sustain the fish for weeks or longer. The risk of this strategy is that misjudging the size of the prey, consuming something that is too large to digest before it begins to decompose, can lead to the production of gases in the distended stomach that force the fish to the surface, where it dies. Black swallowers found floating dead at the ocean surface often have a partially digested fish larger than themselves, visible through their distended, translucent abdominal wall. The strategy is not without hazard, but the alternative, hunting only prey small enough to present no such risk, may not provide enough calories to survive in the midnight zone. Evolution in the midnight zone selects for strategies that work often enough to produce surviving offspring, not strategies that are safe. The fang-toothed fish, Anoplogastra cornuta, holds a record that has nothing to do with size and everything to do with proportion. The fang-tooth has the largest teeth relative to body size of any fish in the ocean. Its lower jaw fangs are so long that, as with the viperfish, the fish cannot close its mouth fully. Instead, the lower teeth fit into specialized sockets in the upper jaw when the mouth is shut, to prevent them from penetrating the fish's own brain. The fangtooth is typically 16 centimeters long. Its fangs are long enough that a scaled-up version the size of a human being would have teeth the length of a human forearm. These teeth are not for display. They are functional weapons in a world where prey is scarce and every encounter with potential food must be converted into a meal, if at all possible. A prey item that touches those teeth is not going to escape. The mechanical advantage they provide, the leverage of a long tooth applied at its tip, is extraordinary. The fangtooth does not rely on speed or maneuverability, neither of which is particularly developed. It relies on the certainty that once it has made contact with prey, the prey is not getting away. This is a reasonable strategy in an environment where prey encounters are infrequent and where the energy expended in pursuit must be recovered from the prey itself. The predator-prey dynamics of the midnight zone operate within an energy budget that is far more constrained than anything at the surface. Food arrives from above as marine snow, as the bodies of organisms that died in the mesopelagic zone and sank, as the downward migrations of organisms making daily vertical migrations and occasionally not returning. This supply is unpredictable in both quantity and timing. The hunters of the midnight zone have therefore evolved strategies that minimize energy expenditure between feeding events, that maximize the caloric return from each successful hunt, and that allow them to survive extended periods without food. Most midnight zone predators are ambush predators rather than pursuit predators, waiting for prey to come within striking distance rather than actively chasing it, because waiting costs almost nothing while pursuit is energetically expensive. Most have metabolic rates that are fractions of what comparable surface-dwelling fish require, allowing them to sustain themselves on food supplies that would starve a surface fish of the same size in days. And most have bodies that are optimized for the conversion of food into stored energy, with fat reserves that can sustain the animal through lean periods of days or weeks. The hunters of the midnight zone are not merely formidable. They are patient, and patient in the midnight zone is the most important thing to be. The deep-sea gulper eel, Uripharynx pelicanoides, represents perhaps the most dramatic expression of the midnight zone's approach to the feeding problem. The gulper eel is primarily a long, thin, eel-like body with a mouth attached. The mouth is enormous, disproportionate to the rest of the animal in a way that seems almost physically impossible. It can open to an angle of 180 degrees, creating a bag of skin and bone that is larger than the rest of the eel's body. The jaw has no teeth of consequence. The gulper eel does not bite its prey and hold it. It engulfs them, opening its enormous mouth and sweeping through concentrations of prey like a net, swallowing whatever enters the opening before it can escape. At the end of its tail, the gulper eel carries a small bioluminescent organ that glows pink and red, an apparent lure that attracts prey toward the tail while the eel's body hangs in the water column. Prey attracted to the glowing tail swim toward it and into the neighborhood of the open mouth. The eel pivots and engulfs them. It is a hunting strategy built around the geometry of the mouth rather than the physics of pursuit, a solution to the problem of feeding in the dark that requires almost no energy expenditure and that can capture prey of a wide range of sizes with a single movement. The Pacific viperfish's daily vertical migration connects the midnight zone to the mesopelagic zone above it. and illustrates the way in which the midnight zone's hunters are not confined to a single depth, but range across depth. Zones, in ways that link the food webs of different zones together. Many bathypelagic predators migrate upward into the mesopelagic at night, feeding in the relatively denser concentrations of prey available there before descending again at dawn. This migration is not the massive, biomass-dominated migration of the mesopelagic itself, involving billions of small organisms. It is a predator migration, a much smaller in scale but ecologically significant movement of larger animals between depth zones in search of food. The viperfish descends to the midnight zone during the day to avoid the larger predators that move through the mesopelagic in daylight, and ascends at night to exploit the prey that have come up from below. The midnight zone is therefore not simply a habitat, but a refuge, a daytime safe zone for animals that exploit the mesopelagic at night, and the ecological boundary between the midnight zone and the twilight zone above is permeable in both directions, and both directions matter. The colossal squid, Mesonychoteuthis hamiltoni, deserves its own attention as a midnight zone predator because it differs from the giant squid in ways that are ecologically significant. The colossal squid is heavier than the giant squid, though not necessarily longer. Its arms and tentacles are shorter relative to its mantle, but armed with rotating hooks rather than sucker rings. These hooks can rotate freely on a ball and socket joint, allowing them to grip and tear flesh rather than merely holding prey. The colossal squid lives primarily in Antarctic waters, at depths of 1,000 to 2,200 meters, in some of the coldest and most food-limited ocean in the world. Among its documented prey items is the Patagonian toothfish, also known as Chilean sea bass, a large, commercially valuable fish that shares its depth range, though the full range of its diet remains incompletely known. The colossal squid's encounter with the sperm whale is parallel to the giant squids, documented in the same indirect ways. Beaks in stomachs, hooks embedded in whale skin, acoustic patterns suggesting hunting behavior. The two largest invertebrate predators in the midnight zone occupy different ocean basins and different depth niches, but face the same fundamental challenges and have arrived at broadly similar, though not identical, solutions. The midnight zone, in different parts of the world's oceans, has independently produced predators of comparable scale and comparable capability, because the midnight zone everywhere presents the same fundamental selection pressures. The predatory strategies of the midnight zone have something to teach about evolution more broadly. The midnight zone has been a stable environment for tens of millions of years, a place where the fundamental physical conditions, darkness, cold, pressure, scarcity, have changed relatively little compared to the dramatic shifts in temperature and chemistry that have characterized the surface ocean over geological time. This stability has allowed the arms race between predator and prey to run for an extraordinarily long time without the external disruptions that periodically reset the evolutionary trajectories of surface organisms. The result is predatory adaptations of a refinement and specificity that surface environments more subject to climate fluctuation and physical disruption have rarely produced. The dragonfish's private wavelength, the stoplight loose jaw's missing jaw membrane, the anglerfish's parasitic male, These are the products of evolutionary fine-tuning that has had millions of uninterrupted years to work. They are not crude approximations of a solution. They are the solution, optimized over timescales that dwarf the entire history of human technology. And below the zone where these hunters operate, in the biological economy that sustains them without their awareness, a different kind of story is unfolding. Every organism in the midnight zone ultimately depends on something it has never seen and never will see: the productivity of the sunlit ocean thousands of meters above. The darkness of the midnight zone is not self-sustaining. It is subsidized, continuously and from a distance, by the photosynthesis occurring in the sunlit zone above, where phytoplankton converts sunlight and carbon dioxide into organic matter that enters the food web and eventually, through a series of deaths and sinkings and decompositions, reaches the midnight zone as the faint, continuous precipitation of organic particles that oceanographers call marine snow. Marine snow is the economic foundation of the midnight zone. Without it, the hunters and the hunted and the bacteria that process what the hunters leave behind would have nothing to sustain them. The midnight zone is not independent of the surface. It is the surface's downstream, the place where the surplus production of the sunlit ocean, everything that was not consumed in the layers above, eventually arrives. Marine snow is not a uniform material. It is a heterogeneous mixture of organic particles of widely varying size, composition, and nutritional value. The smallest particles are individual bacterial cells and fragments of dissolved organic matter that have aggregated into loose flocks. The larger particles include the fecal pellets of zooplankton, the mucus-feeding webs of larvaceans that have been shed and are sinking, the molted exoskeletons of crustaceans, the bodies of phytoplankton cells that have died and clumped together, and fragments of larger organisms that died in the water column above. The particles sink at rates that depend on their size and density, from a few meters per day for the smallest and least dense particles to hundreds of meters per day for dense fecal pellets and aggregates. The slowest particles may spend weeks or months in transit between the surface and the midnight zone, during which time they are progressively consumed by bacteria and other organisms at intermediate depths. By the time the remaining particles reach 1,000 meters, the amount of organic carbon they represent is typically only 1 to 5% of what left the surface. The midnight zone receives not the full output of surface photosynthesis, but its residue, the fraction that survived the gauntlet of consumption in the water column above. The particle flux that reaches the midnight zone is not uniform in space or time. It is concentrated in areas of high surface productivity, beneath the great blooms of phytoplankton that develop in nutrient-rich coastal waters and in upwelling zones. It is more abundant in spring and summer, when surface production is highest, than in autumn and winter. It is episodic, arriving in pulses associated with bloom events and with a sinking of large organisms, rather than as a constant, steady drizzle. The organisms of the midnight zone have adapted to this episodic supply in the same way that desert organisms have adapted to episodic rainfall, by being able to exploit sudden abundance rapidly and to survive the inevitable periods of scarcity that follow. The metabolism of midnight zone bacteria can increase many times over in response to a pulse of fresh organic matter, then return to near dormancy when the pulse is exhausted. The amphipod crustaceans that scavenge the seafloor and the water column can swarm in enormous numbers over a newly arrived food source and then disperse when it is gone. The patience that characterizes the predators of the midnight zone extends to every level of the food web. The whale fall is the most dramatic example of episodic food delivery to the midnight zone, and it represents one of the most consequential single events in deep-sea ecology. When a large whale dies in the open ocean, its body begins to sink. A carcass the size of a blue whale, up to 150 tons of organic matter, descends through the water column over a period of days, arriving on the seafloor at depths that may include the midnight zone or the abyssal zone below it. The descent of a whale carcass is not a passive fall. It is an active ecological event. Mobile scavengers, primarily sleeper sharks and hagfish, begin feeding on the carcass within hours of its sinking, consuming the soft tissues over a period of weeks to months in what ecologists call the mobile scavenger stage of whale fall succession. The bones remain after the soft tissue is gone, and they harbor a community of organisms during what is called the enrichment opportunist stage, in which polychaete worms, crustaceans, and other invertebrates colonize the bone surface and consume the lipids and proteins trapped within. The final stage, the sulfophilic stage, occurs as bacteria begin to break down the lipids in the whale bones through a process of anaerobic decomposition that produces hydrogen sulfide, the same toxic gas that sustains hydrothermal vent communities, and that here sustains a chemosynthetic community around the whale bones that can persist for decades after the whale's death. A single whale fall can sustain a diverse biological community for 50 to 100 years. It is a temporary island of productivity in the food desert of the deep sea, and the organisms that colonize it have evolved specifically to exploit it. The vertical migration of mesopelagic organisms represents the active component of the biological pump, the living conveyor belt that moves carbon and nutrients from the surface to the deep in the bodies of organisms rather than in sinking particles. Every evening, billions of small fish, squid, shrimp, and other organisms ascend from depths of 200 to 1,000 meters toward the surface, where they feed on the phytoplankton and zooplankton of the sunlit zone. Before dawn, they descend again. During their time at depth, they excrete, respire, and in some cases die and sink, transferring the carbon they consumed at the surface into the deep ocean. This active transport supplements the passive sinking of marine snow and represents a significant fraction of the total biological pump, perhaps 10 to 40% of the total carbon flux in some ocean regions. The organisms that make this migration are not doing it to move carbon. They are doing it to feed and to avoid the predators of the surface layer during daylight. The carbon transport is an inadvertent consequence of their behavior. But inadvertent or not, it is one of the most important biological processes in the ocean, connecting the surface to the deep through the metabolism of billions of small bodies making the same journey every night. The fecal pellets produced by zooplankton in the surface ocean are one of the most efficient mechanisms for transporting organic carbon to the midnight zone. A fecal pellet, compact and enclosed in a membrane that slows decomposition, sinks much faster than the loose organic particles that constitute the bulk of marine snow. Large copepods can produce pellets that sink at 200 meters per day or more, carrying carbon from the surface to the midnight zone in a matter of days rather than weeks. The nutritional value of these pellets, while lower than fresh phytoplankton, is sufficient to sustain the bacteria and other organisms that intercept them in the water column and on the seafloor. The production of fecal pellets by zooplankton is therefore a critical link in the chain, from surface photosynthesis to deep-sea biology, and anything that affects zooplankton populations or behavior, changes in surface temperature, acidification, changes in the species composition of phytoplankton communities, can alter the efficiency of this link and change the amount of carbon that reaches the midnight zone. The deep-sea sediment that underlies the midnight zone is not merely the final destination of the biological pump. It is an active participant in the cycling of carbon and nutrients. The bacteria that live in the sediment, in concentrations that can reach millions of cells per cubic centimeter in organic-rich areas, continue to process the organic matter that arrives from above long after it has settled. This processing releases carbon dioxide, which dissolves in the deep water and contributes to the chemistry of the ocean. It releases nutrients, nitrogen, and phosphorus compounds that are eventually carried upward by the thermohaline circulation and returned to the surface, where they fuel future photosynthesis. The sediment is not a dead archive. It is a slow-motion chemistry laboratory, running reactions that began when the organic matter arrived, and that will continue for years or decades until the material is fully processed. The sediment of the midnight zone connects the biology of the present to the geology of the past, and the chemistry it produces shapes the biology of the future. The carbon that the midnight zone processes and stores is not trivial in its implications for the global climate. The deep ocean, including the midnight zone and the abyssal zone below it, contains approximately 38,000 gigatons of dissolved inorganic carbon, more than 40 times the amount currently in the atmosphere. The biological pump that delivers organic carbon to the deep removes carbon from the atmosphere and stores it in the deep ocean on timescales of centuries to millennia, depending on how long the deep water circulation takes to return it to the surface. Without the biological pump, atmospheric carbon dioxide concentrations would be dramatically higher than they currently are, and the climate of the Earth would be correspondingly warmer. The midnight zone is not merely a biological curiosity. It is a critical component of the global carbon cycle, a mechanism by which the productivity of the surface ocean moderates the chemistry of the atmosphere. What happens to the biological pump as the ocean warms and acidifies is one of the most consequential unanswered questions in Earth science, and the answer will be determined in large part by what happens in the midnight zone. The biological pump operates differently in different parts of the midnight zone. In the upper midnight zone, where the flux of organic matter from above is still relatively high, and the temperature is still several degrees above freezing, bacterial activity is brisk enough to process most of the arriving organic matter within a relatively short time. In the lower midnight zone, where the flux has been further reduced by consumption in the layers above, and the temperature approaches freezing, bacterial metabolism slows and organic matter may persist for longer before being fully processed. The gradient in biological activity from the top to the bottom of the midnight zone mirrors the gradient in the flux of organic matter from the surface, creating a depth-structured biological economy in which the richest communities are at the top, closest to the source of food, and the most food-limited communities are at the bottom, where the particles that have survived the entire journey through the water column finally come to rest. This gradient is not smooth. It is interrupted by topographic features, by seamounts and ridges and abyssal hills that concentrate currents and food in ways that create local patches of elevated productivity in an otherwise sparse environment. The role of the midnight zone in the nitrogen cycle is as consequential as its role in the carbon cycle, though less widely discussed. Nitrogen is an essential element for all life, required for the construction of proteins and nucleic acids. In the ocean, the availability of nitrogen in biologically usable forms, primarily nitrate and ammonium, limits photosynthesis in most ocean regions. The nitrogen that fuels surface photosynthesis ultimately comes from two sources: nitrogen fixation by specialized bacteria and cyanobacteria that convert atmospheric nitrogen into biologically usable forms, and the upwelling of nitrate from the deep ocean. The nitrate in the deep ocean is the product of the decomposition of organic nitrogen, the proteins and nucleic acids of organisms that died in the surface ocean, and whose remains were decomposed by bacteria in the water column and sediment, releasing the nitrogen they contained back into the water as ammonium and then nitrate. The midnight zone is where much of this decomposition and nitrogen regeneration occurs. The nitrate that upwells to the surface to fuel tomorrow's phytoplankton bloom was, weeks to centuries ago, the protein in a copepod or a fish or a phytoplankton cell that died in the surface ocean and sank. The midnight zone processes that protein and returns the nitrogen. The ocean's nitrogen economy, like its carbon economy, passes through the midnight zone. The hydrothermal vent systems that exist at the mid-ocean ridges, which run through the midnight zone at depths of 2,000 to 4,000 meters, represent an input of energy to the midnight zone that is independent of the surface and independent of the sun. The vents release hydrogen sulfide and other reduced chemical compounds at temperatures up to 400 degrees Celsius, and chemosynthetic bacteria use these compounds as an energy source, in the same way that photosynthetic bacteria use sunlight. The communities that form around hydrothermal vents, the tube worms and clams and shrimp and crabs that depend on chemosynthetic bacteria either directly or through symbiosis, are entirely independent of the marine snow economy that sustains the rest of the midnight zone. They are running on geological energy rather than solar energy. This independence from the sun was what made their discovery in 1977 so revolutionary. It demonstrated that life on Earth did not universally depend on solar energy, and that entirely alternative energy sources could sustain complex biological communities in the deep ocean. The hydrothermal vent communities are minority communities in the midnight zone. The vast majority of the midnight zone's biology runs on marine snow on the diluted and degraded remnants of surface photosynthesis. But the vents are a reminder that the rules of the surface world, including the rule that energy ultimately comes from the sun, are not universal. The midnight zone has always known things about biology that the surface world is still learning. The oxygen minimum zone, which occupies a layer within and just below the mesopelagic zone in many parts of the world's oceans, plays a crucial role in the nitrogen cycle of the midnight zone and illustrates the way in which the physical and chemical structure of the water column shapes the biology of the deep ocean. In the oxygen minimum zone, where dissolved oxygen drops to near zero, a community of anaerobic bacteria thrive by using nitrate as an oxidant in the absence of oxygen. This process, called denitrification, converts nitrate back to nitrogen gas, removing nitrogen from the biologically available pool and returning it to the atmosphere. Denitrification in oxygen minimum zones is one of the primary mechanisms by which the ocean loses fixed nitrogen, and it represents a significant drain on the nitrogen economy that sustains surface photosynthesis. The rate of denitrification in oxygen minimum zones is sensitive to changes in oxygen content, which is in turn sensitive to changes in ocean temperature and circulation. As the ocean warms and oxygen minimum zones expand, the rate of denitrification may increase, reducing the availability of nitrogen in surface waters and potentially limiting photosynthesis and biological pump efficiency. The midnight zone's chemistry is not separate from the surface world's productivity. It is one of the variables that determines it. What the biological economy of the midnight zone ultimately reveals is the depth of the connection between the ocean's layers. The surface produces, the water column processes, transforms, and transfers. The midnight zone receives, processes further, and stores. The sediment archives what the water column above has provided, keeping records that span millions of years of ocean history. These are not separate systems. They are stages in a single planetary scale process. The cycling of carbon and nitrogen and other elements through the ocean and the atmosphere, a process that has been running since the ocean first formed more than 4 billion years ago, and that will continue until the sun becomes too bright for liquid water to exist on Earth's surface. The midnight zone is not the end of this process. It is the middle, the slow and dark and cold middle, where the real work of the ocean's chemistry happens, where the organic becomes inorganic and the inorganic is returned, eventually, to the surface to become organic again. The midnight zone does this work without fanfare, without light, without speed, in the cold and the pressure that the surface world cannot enter, without extraordinary preparation. And it does this work on a scale and with a thoroughness that no surface process can match. What we have barely begun to understand about this work, and what the midnight zone still holds that we have not yet found, is a question that the darkness below has been keeping to itself for a very long time. How much of the midnight zone have we actually seen? The question sounds simple. The answer is humbling. The midnight zone occupies a volume of ocean that spans the entire global ocean basin at depths between 1,000 and 4,000 meters. A volume so large that every direct human observation of it, every dive by every submersible ever built, represents a vanishingly small sample of the total. The total area of the seafloor within the midnight zone depth range is approximately 300 million square kilometers. The total area that has been imaged at high resolution by submersibles and ROVs is estimated at a few thousand square kilometers at most. The fraction of the midnight zone's volume that has been directly sampled, visited, or observed in any way is a number that approaches zero. We have inferred the rest from the indirect evidence available at the surface, the acoustic signals that deep-sea organisms produce, the chemistry of water samples collected on cables, the organisms that come up in nets, the sediment cores extracted from the floor below. These inferences are not worthless. They have given us the broad outlines of the midnight zone's biology and physics. But outlines are not maps, and maps are not the territory. The territory of the midnight zone remains, in almost every meaningful sense, unknown. The species diversity of the midnight zone is one of the most consequential unknowns in biology. Estimates of the total number of species in the deep ocean vary enormously, from tens of thousands to millions, reflecting the genuine uncertainty in our sampling. The midnight zone appears to be diverse by the standards of deep environments, with different species assemblages at different depths, in different ocean basins, over different substrate types, in the vicinity of hydrothermal vents and away from them. Every expedition that targets a new depth or a new location returns with species that have never been described. The rate of new species discovery in the midnight zone has not declined despite decades of deep-sea research. If anything, as sampling technology has improved and more expeditions have reached deeper targets, the rate of discovery has accelerated. The inventory is nowhere near complete. It may never be complete, given the scale of the environment and the difficulty of sampling it. But what is already known is extraordinary enough to give a sense of what the rest might contain. The pharmaceutical potential of midnight zone organisms is one of the more practically consequential areas of deep-sea research, and one that has attracted significant scientific and commercial interest. The organisms of the midnight zone, shaped by millions of years of evolution in extreme conditions, have developed biochemistries that have no equivalent in surface organisms. The enzymes that function at midnight zone temperatures, near freezing, are structurally different from the enzymes that function at surface temperatures. They are cold-adapted, maintaining their flexibility and activity at temperatures that would make a surface enzyme rigid and inactive. These cold-adapted enzymes have applications in industrial processes where low-temperature reactions are desirable, including food processing, medical diagnostics, and molecular biology techniques. The polymerase chain reaction, the PCR technique that became the foundation of modern molecular biology, and that has been used in everything from forensic analysis to COVID-19 testing, depends on heat-stable enzymes originally isolated from a hot spring organism. Cold-stable enzymes from midnight zone organisms may prove equally foundational for future technologies. Beyond enzymes, midnight zone organisms have developed novel chemical compounds that defend against predation, inhibit competing organisms, or serve other biological functions in ways that have no precedent in surface chemistry. The search for novel antibiotics, anti-cancer compounds, and other bioactive molecules from deep-sea organisms, has produced a growing list of candidates, several of which have entered clinical trials. The chemical diversity of the deep ocean, representing hundreds of millions of years of evolutionary experimentation in conditions that differ radically from the surface, is a resource of incalculable potential value, and it is almost entirely unexplored. The fraction of midnight zone organisms that have been studied for their biochemistry is smaller than the fraction of midnight zone space that has been visually surveyed. The pharmacological frontier of the midnight zone has barely been identified, let alone explored. The technological development that has done most to expand our understanding of the midnight zone in recent decades is not the submersible, but the autonomous underwater vehicle, the AUV. Unlike submersibles, which require a human crew and have limited endurance, AUVs can be programmed to follow complex three-dimensional trajectories through the midnight zone, collecting data continuously over periods of hours or days, covering far more ground than any crewed vehicle. Unlike remotely operated vehicles, which are connected to a surface ship by a cable that limits depth, range, and maneuverability, AUVs operate independently, returning to the surface when their mission is complete and uploading their data. The latest generation of AUVs can reach full ocean depth, withstanding pressures exceeding 1,000 atmospheres, and can carry a suite of sensors measuring temperature, salinity, oxygen, bioluminescence, acoustic backscatter, and chemical signatures. They have mapped seamounts and ridges and hydrothermal fields in detail that previous technology could not achieve. They have tracked the distribution of bioluminescent organisms through the midnight zone over deal cycles, revealing the daily patterns of light production in a way that fixed instruments and occasional submersible dives could not. They have measured the oxygen minimum zones with a spatial resolution that has allowed researchers to understand the three-dimensional structure of these zones and how they vary with season and climate. The AUV is not merely a better tool for doing what submersibles did. It is a different kind of tool, enabling a different kind of science, asking questions that could not previously be asked. The acoustic monitoring of the midnight zone, primarily through arrays of hydrophones deployed on the seafloor or moored in the water column, has produced some of the most surprising discoveries in deep-sea science. The SOSUS network, originally built by the United States Navy during the Cold War to detect Soviet submarines, has been partially declassified and made available to researchers, giving scientists access to decades of acoustic recordings from the North Atlantic and North Pacific. These recordings have revealed sounds of biological origin that were previously unknown, including the calls of blue whales, which were too low in frequency to be detected by conventional marine mammal survey methods, and a repeated broadband sound called the bloop, recorded in 1997 and for years unidentified, that was eventually determined to be the sound of ice quakes, the cracking of large ice masses in the Antarctic. The hydrophone networks have also detected the acoustic signatures of hydrothermal eruptions on mid-ocean ridges, submarine landslides, and other geological events in the midnight zone that leave no trace at the surface. The midnight zone speaks continuously in sounds too low and too distant for human ears to detect without instruments. The hydrophone networks are listening, and what they hear continues to expand the inventory of what happens in the dark. The climate change implications of the midnight zone are becoming increasingly clear and increasingly urgent. The biological pump, which depends on the health and productivity of the surface ocean, is sensitive to changes in surface temperature, ocean chemistry, and the species composition of surface communities. As the ocean warms, stratification increases, reducing the mixing that brings nutrients from the deep to the surface, and potentially reducing surface productivity and therefore the flux of organic matter to the midnight zone. As the ocean acidifies, the calcium carbonate shells of the foraminifera and coccolithophores that make up a significant fraction of marine snow dissolve before reaching the deep, reducing the carbon flux. As oxygen minimum zones expand in response to warming, the denitrification that occurs within them removes more fixed nitrogen from the system, potentially further limiting surface productivity. These changes are not speculative. They are being measured in the ocean right now, in ongoing monitoring programs that have been running for decades. The direction of change is clear. The magnitude and the speed of the consequences are what remain uncertain. The deep scattering layer, the mesopelagic biomass whose daily migration is one of the primary active components of the biological pump, is itself responding to climate change in ways that are difficult to predict. As surface temperatures rise, the organisms of the deep scattering layer may shift their depth distributions, their geographic ranges, and their migration timing. The predators that depend on them may follow. The prey that depend on them staying may be left behind. The ecological relationships that have structured the midnight zone's food web for millions of years are being stressed by changes that are occurring on decadal rather than geological timescales. Evolution cannot keep up. Behavior and distribution can shift, and some are shifting. But the deeper changes to biochemistry, to reproductive timing, to the fundamental physiological tolerances of deep-sea organisms require evolutionary time. And evolutionary time in the midnight zone is measured in millions of years. The climate is changing in decades. The gap between those two timescales is where the risk lives. The midnight zone's relationship to astrobiology, the study of the potential for life elsewhere in the universe, is one of the more unexpected dimensions of its scientific significance. The discovery that complex biological communities can exist in complete darkness, sustained by chemical energy rather than solar energy, fundamentally changed the criteria that astrobiologists use for assessing the habitability of other worlds. Before the discovery of hydrothermal vent communities in 1977, the conventional assumption was that life required sunlight, and therefore that habitable environments were restricted to planets and moons at the right distance from their host star to receive sufficient solar radiation. The midnight zone demonstrated that this assumption was wrong. If life can thrive in absolute darkness, sustained by chemical reactions between hot rock and cold water, then the list of potentially habitable environments in the solar system expands dramatically. The icy moons of Jupiter and Saturn, Europa and Enceladus in particular, are thought to harbor liquid water oceans beneath their frozen surfaces, kept liquid by tidal heating from their parent planets. If those oceans have hydrothermal activity on their floors, they may have the same combination of liquid water, chemical energy, and minerals that sustains life in the midnight zone of Earth's ocean. The midnight zone is not only telling us about Earth's biology, it is telling us where to look for biology elsewhere. The microbes of the midnight zone are the aspect of its biology that is simultaneously the most important and the most thoroughly overlooked in popular accounts. The midnight zone is dominated by sheer numbers and by metabolic activity, not by the anglerfish and the giant squid and the sperm whale that capture the imagination, but by single-celled organisms too small to see without a microscope. A milliliter of midnight zone water may contain thousands of bacterial cells. A cubic centimeter of midnight zone sediment may contain millions. These microbes are the primary processors of organic matter in the midnight zone. They are the organisms that break down the marine snow, releasing the carbon and nutrients that more complex organisms depend on. They are the organisms that cycle nitrogen and sulfur and iron through the chemical forms that the rest of the food web can use. They are the chemosynthetic communities at hydrothermal vents. They are the sulfate reducers in the sediment. They are the methane producers and methane consumers in the cold seep environments that dot the continental margins. The midnight zone's microbes are doing most of the work. The charismatic megafauna, the hunters, and the glowing creatures that populate the documentaries are the visible surface of an invisible microbial economy that is orders of magnitude larger in biomass and in metabolic significance. Understanding the midnight zone means, first and foremost, understanding its microbiology, and its microbiology has barely been sampled. The connectivity of the midnight zone across ocean basins is a dimension of its ecology that has only recently begun to be appreciated. For a long time, the midnight zone was thought of as a collection of isolated environments, each basin and each trench harboring its own distinct community of organisms. This view has been substantially revised by genetic studies that have revealed connectivity between midnight zone populations across distances of thousands of kilometers. Some midnight zone species have global distributions, their larvae or propagules dispersed by the deep ocean currents that connect the basins. Others show regional differentiation, with populations in different ocean basins that are genetically distinct but clearly related, having diverged from a common ancestor after a period of connection that ended when the circulation patterns changed. The midnight zone is not a collection of isolated ecosystems. It is a globally connected system, linked by the slow circulation of deep water that carries organisms and genes across the ocean floor on timescales of centuries. The connectivity has implications for how we think about conservation. Protecting a midnight zone community in one basin from the impacts of deep-sea mining may not be sufficient if the population is demographically connected to another basin where mining is occurring. The midnight zone is too interconnected for piecemeal protection to be effective. What would it take to genuinely understand the midnight zone? Not in outline, but in detail. Not by inference, but by observation. Not in the way we currently understand it, but in the way we understand the surface ocean, with continuous monitoring, with high-resolution mapping, with systematic biological surveys, with the ability to observe organisms in their natural environment over ecologically meaningful timescales. It would take a sustained, international, multi-decade commitment of scientific resources on a scale that has not yet been attempted. It would take a fleet of AUVs and gliders and landers deployed throughout the global ocean midnight zone, collecting data continuously and transmitting it to surface buoys and satellites in real time. It would take a biological sampling program that goes beyond the trawl net, that collects organisms at depth in pressure-maintaining containers that preserve their condition for physiological study at the surface. It would take a chemical monitoring program that tracks the biogeochemistry of the midnight zone across space and time with the same density of measurements that we apply to the atmosphere. None of this is impossible. The technology largely exists or is within reach. What has been missing is the recognition that the midnight zone is important enough to warrant this investment. That understanding it is not a luxury of scientific curiosity, but a necessity of planetary management. The midnight zone processes the carbon that regulates our climate. It stores the nutrients that feed the ocean's productivity. It contains more of life's diversity than anywhere else on Earth. And we barely know it exists. That is the void at the center of the midnight zone story. Not the darkness of the water, but the darkness of our knowledge. And into that darkness, every expedition descends a little further. Let that thought settle now, as you drift toward sleep, into the cold and the dark and the quiet that the midnight zone knows so completely and so patiently. You are a surface creature, warm and lit, breathing air that the phytoplankton of the sunlit zone produced. But below you, thousands of meters down, the midnight zone is doing its work, as it has always done, in the dark, without haste, without fanfare, sustaining the ocean and the atmosphere and the climate that makes your life possible, utterly indifferent to whether you know it is there. Now you know, and that changes something, even here, even in the quiet before sleep. Rest now. The midnight zone is patient. It can wait. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting.